Let's get started. So I'm very pleased to, maybe let's wait for a few people to tune in. All right, so I'm very pleased to introduce Dr. Vinod Kumar as our speaker today. Vinod is PhD in neuroscience from Max Planck, Tübingen, from the Institute of Biological Cybernetics, which is a very famous institute. The people here whom I know, colleagues here. He is now working as a postdoctoral fellow there at the institute, as well as a visiting scientist at Donders in Netherlands. And at Donders, you work with Christian Beckman as well. So Vinod spent his whole PhD life looking at this one structure called the thalamus, and it's a very nice paper describing the functional anatomy of the thalamus. So Vinod here in the US is attending a conference exclusively for thalamus in Ventura. I don't know if you know that. And so it was very kind of you to actually come here and talk to us about your findings.
Good afternoon, dear ladies and gentlemen, and thank you for the kind introduction. I would like to thank Shantanu, Ashish, and Johanna for the invitation. And so it is, okay, please come. It is an immense pleasure to talk about our work. I will present the past and ongoing work which is focused on the structural and functional anatomy of the thalamus. And since Shantanu mentioned that we were in the conference, so I was organizing first part of the conference, and yesterday we had a party, so I was awake until 2 a.m., so a little bit hangover. So if you miss something, please ask. It could be that something I forget to mention or something. So before I start, I would like to acknowledge Professor Wolfgang Groth, Christian Beckmann, and Klaus Scheffler for their support and supervision, and for technical support, and also our supercomputing facility at Donders, at Max Planck, and also in the clinic, and also for the funding by the German Science Academy. In my presentation, I will start with the anatomy and function of the thalamus, and subsequently I will present results on the structural and functional parcellation of the thalamus. And afterwards, I will show some results on the thalamo-cortical structural connectivity and cortico-thalamic functional connectivity. Thalamus is a central connectivity hub in our brain. It lies in the center of the brain, as shown in this mid-sagittal view of the human brain. The colors doesn't look so nice as in my screen, but so the different colors indicate differences in cytoarchitecture of cortex and of subcortical areas. The significance of the thalamus can best be understood by looking at connectivity patterns of the brain, as shown here. So the tracts project from the brainstem and the cerebellum to the thalamus, and via the thalamus to different cortical areas. For example, you see projection to the visual cortex, parietal, sensory, motor, and to the frontal cortices. Almost all input to the cortex pass through the thalamus, and therefore thalamus acts as the switching and integration center of the brain and serves as a gateway to the cortex, and involves in attention, arousal, awareness, memory, voluntary movement, and other functions. A closer look on the thalamocortical connectivity pattern is given in this schematics drawing by Professor Neuenhaus, in which you see different thalamic nuclei and their specific cortical projections. And on the right side, you see a 3D scheme of the thalamus, which depicts different thalamic nuclei and displays different compartments, which project to different cortical areas. For example, medial thalamus project to sensory and motor areas, anterior to the limbic system, medial to the frontal cortices, and posterior mainly to the auditory and visual areas. And in addition, you also have number of intralaminar nuclei which facilitate rapid communication between brainstem and the cortex. So major question of our study was, can we differentiate thalamic nuclei using magnetic resonance imaging? In this slide, I have summarized MRI studies of the thalamus using different conventional imaging modalities. So, for example, on the left you see postmortem MRI at 4.7 Tesla. On the right side, magnetization transfer MRI at 3 Tesla. And same brains T1 at 1.5 T and 4 Tesla. And even susceptibility MRI at 7 Tesla. And you can see that all modalities exhibit only a limited contrast, and therefore differentiation of different thalamic nuclei is not possible. One good exception is a nice contrast in the pulvinar nuclei in the susceptibility MRI due to enhanced vascularization. And again, so in summary, so conventional MRI does not provide a sufficient tissue contrast, and you cannot see thalamic nuclei in detail. And that brings us to diffusion imaging. So diffusion imaging is a different kind of MR imaging modality which is able to depict microstructural differences in more detail. Using diffusion imaging, one of the first study was published by Voxel and colleagues in 2003. She performed diffusion imaging using 6 diffusion directions and used k-mean clustering on the diffusion eigenvectors and achieved 14 clusters in the thalamus. She then made comparison of her results with the histology and found no good correspondence. And one of the reasons was k-mean clustering does not account for spatial topography of the clusters, and therefore you see these gradient-like structures which have no clear correspondence with the histology. In the same year, Oxford group manually parcellated the cortex in 7 subdivisions, and they performed DTI tractography using these 7 cortical subdivisions as a start region for direct tracking to the thalamus. As a result, they could distinguish 7 subdivisions in the thalamus. However, again, comparing these connectivity-based segmentation result with the histology, no good agreement was found. Mainly because connectivity-driven segmentation method mainly rely on tractography pathways and does not account for underlying local microstructural properties in the diffusion data. And therefore we thought to perform our own segmentation of the thalamus using another segmentation approach. In the study, we examined 63 volunteers, 32 male, 31 female, at 1.5 Tesla MRI scanner with minimal acquisition parameters. And we performed state-of-the-art preprocessing of that time, and we computed principal eigenvector at each voxel by fitting the diffusion tensor model. We developed a new segmentation method to segment the thalamus by using the orientation of the principal eigenvectors. And in the method, at first we determined 21 equidistant reference orientation on a sphere, and in the next step we assigned each voxel's principal eigenvectors orientation to the closest reference orientation for each subject for right and left thalamus in male and in female group. And finally, we performed comparison of our segmentation results with the histology atlas of the thalamus to determine the anatomical significance of the segmentation. So this slide shows our method in bit in more detail. So on the top you see illustration of the predetermined 21 equidistant orientation in different views. And on the bottom, okay, you don't see clearly. On the bottom you see an axial slice of the thalamus which depicts principal eigenvector at each voxel as well as corresponding fractional anisotropy map, and segmentation results on the right side. So you can see that it is possible to sort different eigenvectors into different clusters. So the sense of the algorithm is that similar oriented voxels may belong to the same nuclei. And the rendered group results are shown here for the male group and for the female group at single subject level, at group level, and at group probability of 50% and 90%. And you can see that method nicely parcellates thalamus in single subject level in male as well as in the female group. And secondly, you can see that there are differences in between left and right thalamus in the cluster organization, but also between male and in female. And these differences persist in the group level in male as well as in the female group. At higher probability of 50% and 90%, only stable clusters remain but shrink in size. So in general, we found approximately 6% laterality differences and 4% gender differences. And especially, so posterior, lateral anterior parcels were left lateralized, and anterior and mediodorsal cluster were right lateralized. In the next step, we took these clusters which survived 90% probability, and we performed DTI tractography in the male as well as in the female group. And the group results are shown here for the male and for the female at probability of 50%. And you can see that these clusters project to different cortical areas. For example, posterior cluster project to visual cortex, lateral posterior to the sensory cortex, I think it's gone, okay. And lateral anterior to the motor cortex, and medial to the frontal cortices. And this is interesting because thalamus is a small structure, and these clusters appear spatially very close, but yet they show specific cortical projections. And even if we constrain probability at 90%, major projections remain, but they shrink in the size. And if we then look at surface projections of these tracts, then we see laterality differences. So, for example, lateral anterior cluster project to the motor cortex. So you don't see clearly, okay. And this is left lateralized in male as well as in the female group, while somatosensory projections are bilateral. So in summary, each cluster possesses specific cortical projection sites. And this slide summarizes our DTI findings. So method permits stable parcellation of thalamic substructure in individual and in group, and it's simple to perform. So it takes like 10 to 15 seconds per subject, and you don't need any prior segmentation of the cortical areas. And clusters showed variability in the size, location, and stability, and we found more laterality differences than gender differences. And clusters have approximate correspondence to histologically defined thalamic nuclei and their cortical connectivity. So this part is mainly written in the paper. So if you want to interested in the histological comparison. Now I want to switch to my next study, in which we performed parcellation of the thalamus using resting state fMRI. So with this introduction, I just want shortly to show how resting state fMRI works, using an example from the study by Reichle and colleagues. You must be aware about it. So, okay. On the top, you see here a set of 125 images acquired at rest without any explicit task condition in a 5-minute fMRI measurement. Now, we can take one voxel of a selected cortical area, extract its time course. And we can take this time course to perform time average correlation with all other voxels in the brain. As a result, we can now see that only the bilateral somatosensory and supplemental motor area possesses similar spatiotemporal pattern. Using similar approach by taking different time courses of voxels in different brain areas, we can depict different functional networks within the brain. Therefore, our question was, can a thalamic parcellation be achieved based on the spatiotemporal similarities in the resting state fMRI? We therefore took the data of 40 volunteers from Human Connectiome project, which were acquired at 3 Tesla with a resolution of 2 mm isotropic. So in total, the data size consists of approximately 192,000 fMRI scans. We performed state-of-the-art preprocessing, and we developed a new method to capture the variance in the fMRI time courses using instantaneous correlation analysis method, which is illustrated in more detail in next slide. We determined the reliable and reproducible number of parcels using split half reproducibility analysis and performed group independent component analysis. So our applied instantaneous correlation analysis method is shown here in more detail. So on the left you see a schematics of the thalamus, which consists of 4 different compartments and their voxel time courses. And the bottom you see mean time course over all the 4 compartments. And then we divided each voxel time course with the mean. And this results in temporal unfolding of the fMRI time courses. And that allows better estimation of sparsity in the ICA. And this resulted unfolded signal were fed into ICA, and we determine the reproducible number of parcels using split half reproducibility analysis. In the split half reproducibility analysis, we randomly did 20 splits of the data, and then we performed ICA from dimensionality of 2, 3, 4 until 50. And in the next step, we looked at spatial overlap of the components within each dimensionality across all the splits. And this is shown here as a violin plot, and you see here mean and median. The colors are not clear, and this is only the median are shown here. And this spatial overlap we understand as a reliability score. So the components which will have more overlap, they are more reproducible in the data. So the idea is that it doesn't matter how many sources are in the data, but how many we can reliably and reproducibly estimate. And this is important because in the clustering algorithm, usually you have major problem how many component you should estimate. And so that's why we use such approach. So what does this plot mean? So you have higher the reliability score, that means the stable the functional configuration is. And this is for right thalamus, so we found 15 parcels showed peak reliability score. And for the left thalamus, 17 parcels showed peak reliability score. And then we visualize the most stable functional configuration, and the results are shown here for the right thalamus and for the left thalamus. So the spatiotemporal similarities revealed 15 parcels within each thalamus. In addition, we also observed that the parcel shows laterality differences. For example, parcel 2 appear at similar location in the right thalamus as well as in the left thalamus, but differs in size and spatial distribution. To get a better overview of the laterality differences, we computed dice overlap of each right parcel with each left parcel. And in the resulting table for the right and left parcels, you can see that there is considerable difference exists. For example, parcel 3 and parcel 5 are only appear on one side. Parcel 8 and 9 have 2 left subparcels, and others also vary considerably. And this is very, very interesting because in thalamic histology, it has been laterality differences has been ignored, and it has been presumed that thalamus possesses mirrored representation in both the hemisphere. And therefore, question arises, why do these differences occur? And everybody knows that brain is functionally lateralized, as it is summarized nicely in the book by Betty Edwards. So there are numerous functions like language, analytical thought, and others. They dominantly use left brain hemisphere, while other functions dominantly use right hemisphere. Therefore, we suggest that the laterality differences of thalamic parcels may be linked to differences in hemispheric functions of cortical areas. In the next step, we performed histological comparison of our functional parcellation, and the results are shown here. Our parcels are depicted in the numbers and histological nuclei in the letters. And so the main message here is that some parcel fit nicely while others show only less overlap. So in principle, there is only rough correspondence with the histology. And one of the reasons behind this is the histological atlas only consists of 2 to 6 brain specimens, and therefore does not account for inter-individual variability. Okay. Finally, at first, we looked at structural cortical connectivity of right and left thalamus parcels using available DTI data in the same set of subjects in HCP. And the group results are shown here for the direct tracking. In the direct tracking, we allowed each parcels to go directly to the cortex without any contribution from the other parcels. And you can see results for, so medial lateral view for the right hemisphere, this is left hemisphere and combined views. So, and you can see that these parcels project to specific cortical areas. And even we don't tell where to go, so still they nicely segregate the cortex. For example, visual cortex, you have temporal lobe, parietal, sensory, motor, and to the frontal cortices. And if we allow parcels to go not only directly but also via other parcels, then spatial configuration slightly changes. And this is shown here. So its color is not very nicely visible. For example, so parcel 3 is now have dominant projections of parcel 1. And this suggests that there are some parcels project via other parcels to the cortex in the DTI data. And in the next step, we looked at cortical functional connectivity of right and left thalamic parcels, and the resulting group fixed effect map is shown here for the right hemisphere and for left hemisphere. And we found that there is a widespread distributed functional connectivity of right and left thalamic parcels. And even when we compute strongest correlation at each voxel, still we see that there is a disseminated connectivity pattern in the cortex. So there are striking difference between the structural and functional connectivity, and these disseminated connectivity pattern may be due to intrathalamic, subcortical, or corticocortical interactions. In the next step, we performed comparison of over structural connectivity results with Allen mouse connectivity atlas. So what Allen Institute did is they injected retrograde tracers in the cortex, and then they looked at their propagation, and these maps are available online. So people have also did a lot of work with monkey like Peter Strick, but these maps are not available in public. So we compared mouse, which was available. And on the left, you see over parcels and their structural connectivity results on the sagittal and coronal view and in the glass map projection. And you can again see that they project to different cortical areas. If we now compare our result with Allen mouse connectivity atlas, then we see that there is a remarkable convergence exists between over structural connectivity maps and Allen mouse connectivity maps. And we also looked at the projection areas, that which area they project, and they are moreover homologous representation, and which indicates that phylogenetic similarities exist even between man and mouse. And in the final step, we compared our functional cortical connectivity results of the intralaminar and midline parcels with the Allen mouse connectivity atlas. And according to our direct DTI results, these parcels do not project directly to the cortex. However, nevertheless, we found widespread functional connectivity with these parcels on the cortex. And again, you don't see actually very well. Okay. Again, comparing these results with the Allen mouse connectivity atlas, we found that there is a very nice correspondence. Okay, this slide summarizes our functional parcellation study at 3 Tesla. The functional parcellation depicted 15 reliable and reproducible parcels within each thalamus. Parcel shows laterality differences and rough correspondence with the histology. Parcel showed specific structural and widespread functional connectivity profiles. The structural and functional connectivity map delineated a remarkable convergence with Allen mouse connectivity maps. And whoever is interested to use these functional parcellation, because it's really built on a lot of data, is available publicly here, and one could exactly apply these maps and investigate individual subjects' corresponding functional anatomy. And here I put one point that further work is necessary to improve the functional anatomy of the thalamus with improved spatial resolution at ultra high field MRI at 70 or 9.40. And one of the questions, why do we need it? So there are several challenges in measuring subcortex MRI. but I only want to mention one important point, and that is as we increase the strength of the magnet, we see more. And this is really important for subcortical nuclei such as thalamus, because there are some nuclei only consists of few thousand cell bodies. And therefore, our question was, can we differentiate more detailed functional anatomy at 70 and 9.40? And we looked this at 70, the data obtained from Human Connectiome project in 62 subjects, and we performed exactly similar parcellation which just I showed you in my previous study. And the results are shown here. For the left thalamus in lateral and medial views, and right thalamus medial and lateral, and these are combined views, superior, inferior, anterior and posterior. The spatiotemporal similarities revealed 38 parcels within each thalamus. And at 9.40, the spatiotemporal similarities revealed 83 right and 82 left parcels. So again, you have combined views, superior, inferior, anterior and posterior. So the question is, what do we learn from such a high dimensional functional parcellation? And we know that thalamic nuclei are composed of several subnuclei. So therefore, can we differentiate functional diversity within major thalamic nuclei, such as LGN? And we know that LGN gets input from retina, superior colliculus, and brainstem, and projects to the visual cortex. And it has a defined layered organization, which is shown by Andrews and colleagues. And interestingly, we found that there are functional subdivisions within the LGN, so which is shown here in a coronal and different coronal slices for the left LGN and for the right LGN. And you can see it better in the zoomed version. So on the right side, you see the histological depiction of the LGN. On the left side, result of our functional parcellation. And you can see that our results do not exactly match with the histology. For example, these contours and, for example, the contours doesn't match. So our functional parcellation appears abstract, but we can say that there are functional subdivisions in the LGN. So before I move to the next part of my presentation, I want to show you one application of such a high dimensional functional parcellation. So at 9.40, we performed a task fMRI study in which volunteers were examined with resting state fMRI and a visual task. In the task, they observed natural landscape pictures. So the task paradigm was like standard block design, eye close, natural pictures, eye close. So basically like this. And then we performed standard univariate GLM. And the results are shown here for the differential contrast of picture minus eye close. And you can see there is a visual cortex is activated, Culcarin Fisher primary visual cortex, and also LGN. In the next step, we took these high dimensional functional anatomy of the thalamus and computed parcel by parcel connectivity in all the subjects. And the results are shown here for the rest, for the resting state. Actually, you don't see a lot of stuff, but okay. Resting state, picture viewing task, and eye close condition. This is for the left thalamus and for the right thalamus. And this is just moreover similar depiction of these connectivity matrices, just graphically here. Okay. So, okay, this is not working. So I just show you. So this is here. So left thalamus resting state, picture viewing task, and eye close condition during the task. And this is for the right thalamus. So here we have two main messages. One is that there is a higher connectivity of the thalamic parcels in the picture viewing task compared to the rest. And this higher connectivity persists even in the eye close condition during the task compared to the rest. And that is very interesting because just because as an experimenter we designed that, okay, if I have 1.5 minute block to close the eye and I think thalamus is shutting down, but it's not happening like this. So there is still higher connectivity. So node has slight variance. This I think is informing something that something more is going on. Okay. Okay, this summarizes our functional parcellation at 70 and 9.40. So one of the main messages is that as the resolution improves, functional similarity detection improves. So the plot shows parcels at 3T, 70 and 9.40. And higher order parcellation enabled functional subdelineation of major thalamic nuclei such as LGN. And this probably also holds true for other thalamic nuclei, for example, pulvinar or subparts of maybe mediodorsal. One has to look, I think. And thalamic Parcels connectivity is higher during visual tasks compared to the resting brain. Now I want to switch to the next study in which we performed thalamocortical structural connectivity with histological defined thalamic nuclei. Here on the top you see histological atlas of the thalamus. The different colors show different thalamic nuclei and also their nuclei names. So we took the data of 730 volunteers from the Human Connectiome project, which were acquired at 3T with a resolution of 1.25 millimeter isotropic. And we performed state-of-the-art pre-processing and performed structural fiber connectivity of each thalamic nuclei. And we summarized all the subjects' data just by computing basically average over all the subjects. And for example, so I have the first results I would like to show you for visual thalamus. So visual thalamus mainly consists of 5 different nuclei. That one is the major nuclei is a lateral geniculate nucleus, and then there is different pulvinar nuclei. So anterior, inferior, lateral and medial pulvinar. So LGN is understood to be first order thalamic nuclei. That means it receives input from the subcortical structures and project to the cortex. And pulvinar nuclei are higher order thalamic nuclei. That means they facilitate cortical functioning by real-time interacting for the cortico-cortical interactions. And the group fiber tracking results are shown here, again in different views, and these colors don't really match, but just I want to so there is some kind of organization here. For example, so lateral geniculate project to visual cortex and also frontal cortex, temporal lobe and also brainstem. The anterior pulvinar mainly go to cerebellum and also to the brainstem. This is okay, it's same color here. So the lateral pulvinar goes to visual cortex and also to the frontal cortex. And interestingly, so inferior pulvinar, this is mainly just remains within the thalamus. And so this actually here you have moreover there is different color here. So this is a projection of the medial pulvinar. So medial pulvinar project to higher visual areas and also to the parietal cortex. So basically you have here dorsal visual stream. So the medial pulvinar projections cover the dorsal visual stream, and it also make connection with the temporal lobe. That means it also has ventral visual stream, and then frontal projections cover lateral visual stream. This suggests that it's probably involved a bit more for the vision compared to other pulvinar nuclei. And in the next step, we looked at intralaminar nuclei in the thalamus, and these nuclei are shown here. So on the top you see axial, then coronal and sagittal projections, as well as their rendered projections. And we have few one of them. So I will just not go in the detail. So, but the main thing is that I really like these nuclei because they are really fascinating. So they reside in this beautiful fibrous internal lamina. So this is nice white matter path, and these are several intralaminar nuclei, and they facilitate rapid communication between brainstem and the cortex, and therefore they are implicated in arousal, vigilance, alertness, sexual processing and numerous other functions. And this has been shown that there is if there is a lesion in the, for example, centrolateral nuclei, the people lose arousal. And the connectivity patterns, they showed mainly specific projections but also overlapping. For example, central median and parafascicular nucleus have shared path, and subparafascicular goes to sensory and motor areas, and centromedian goes mainly to the cerebellum and also the brainstem. and I don't see it here, but centrolateral, it has very nice connection with superior colliculus. So this is very specific here. So the arousal and other functional subsystems connect with the individual intralaminar nuclei. Okay, I will skip the summary. And now I would switch to the last part of my presentation. There we looked at cortico-thalamic functional connectivity with 10 well-defined functional networks. So functional networks are behaviorally plausible correlated spatiotemporal patterns in the brain, and they are numerous functional networks in the brain, and each of these functional networks enable different neuronal processing, such as internal brain states like awareness, learning, memory consolidation, predictive coding, mind wandering, etc., using different functional networks. And cortical functional networks using resting state fMRI were evaluated. For example, one of the first study was published by Beckman and colleague, and he showed 8 cortical functional networks in his paper. In a follow-up study, Stefan Smith and colleagues performed functional analysis as well as also comparison with the task activation studies from 30,000 maps obtained from the brain map database, and they found 10 well-matched cortical functional networks. In a follow-up study, Yu and colleagues showed 7 and 17 cortical functional networks, and after that there has been an explosion in the field of functional parcellation of the cortex. For example, the recent study by Matthew Glasser, he performed high-dimensional functional segregation of the cortex and showed 180 parcels within each brain hemisphere. So there are a lot of parcellation exist, but you have very little understanding about statistical and biological meaning of these parcellations. So one of the parcellation which really interested to me by Stefan Smith, because that has a lot of behavioral and functional relevance, because they looked with the large behavioral studies, the data. So we took these 10 cortical functional networks and we asked 2 questions. Okay, sorry, this was different. Okay, so we were interested in these networks, and why we were interested? Because these cortical networks are not a complete picture of functional networks in the brain, because all incoming and outgoing connection to the cortex use thalamus and rest of the subcortex, and thus cortical functional networks deliver only a partial information and fail to elucidate how the networks act as a system in the brain. Therefore, the thalamic involvement with the cortical network should be investigated. And this statement brings us to our recent study. And in the study, our 2 questions were: how do well-known 10 cortical functional networks communicate to the thalamus at rest? And secondly, can we differentiate thalamic involvement with different cortical functional networks during tasks? In the study, we again took similar 730 subjects, so 329 male, 401 female. In total, data was 3.5 million fMRI scans. And again, data was from Human Connectome project. And we performed state-of-the-art preprocessing, and we did correlation analysis. So basically, Smith 10 correlation networks were taken for the classification of thalamic voxel by performing partial correlation analysis. And then we averaged all the correlation maps, so computed basically group fixed effect map as well as strongest correlation at each voxel in the thalamus by performing winner takes it all analysis. And the analysis flowchart is shown here. So it's really simple. So we did pre-processing as Glasser told, then took cortical resting state networks, looked at connectivity, and then looked at group effect map. And the results are shown here. On the left, you see different cortical functional networks as shown by Stefan and Smith. So you have different functional networks here, for example, medial visual, occipital visual, lateral visual, default mode, cerebellum, and so on, other networks. And on the right side, you see fixed effect map of each cortical functional networks in the thalamus, and that is depicted in 6 different axial slices. So you don't see color again clearly, so this is red color here. And the red color shows strongest correlation with the cortical functional networks in the thalamus. And we found that there is a spatial varying communication exists within the thalamus for different cortical resting state networks, which show distinct and overlapping communication with the cortical networks. We have also looked this data in detail, like how each nuclei is communicating. So this is still work in under progress, but I will show you some of the piece of this study more. So in the next step, we looked at strongest correlation at each voxel, and these results are shown here again for right thalamus and for the left thalamus, and this different color basically showing different networks. And then here we have few observations. For example, if you look cerebellum, so this color is really problem. So there are some network really dominantly use more space than other networks. And secondly, we found that there is ordered organization for the left and right thalamus. Even when you just look like randomly, for example, winner takes all approach. And next, there are laterality differences. And these laterality differences we investigated in more detail by taking these clusters and plotting them and also looking at their difference. And this is shown here, so I will not go in detail, but there are laterality differences with respect to how cortical functional networks of each hemisphere communicate with the thalamus. And in the last in the next step, we looked at histological comparison of these winner takes it all induced clusters, and we found very interesting results. For example, right thalamus and left thalamus show strong overlap with pulvinar nuclei, and that is just exactly what I told you. So visual cortex or visual network use. So pulvinar facilitates cortical functioning of the visual network, and therefore it aligns very well with our understanding. And in contrast, so you have frontoparietal network that mainly employs intralaminar and midline nuclei. And default mode network also communicates with the pulvinar nuclei, but it has much less overlap with other nuclei compared to frontoparietal, which suggests that maybe it's mainly cortico-cortical during the rest and doesn't need a lot of resources from the thalamus. And in the next step, we looked at ranking of cortical communication at resting and in the resting state. And the ranking is done using gradual mode analysis. So what exactly we did is we looked at percent nuclei contribution, and this percent nuclei contribution was done by basically summing of correlation values in each network and divided with sum of the correlation values over all other networks and multiply 100. And in the next step, we basically ranked these gradual modes by summing the percent nuclei contribution of all the nuclei and then basically putting them in ascending order. And the results are shown here. So what these circles tell you is that which network communicate strongest with the thalamus during resting state. So here you see frontoparietal right communicates the strongest, and then you have the ordered organization. So cerebellum communicate the least during the resting state. And this, as we looked at this plot, so one of the next question I had is how does this communication changes in different tasks? And luckily, HCP sample has 7 different tasks, which are motor, language, working memory, emotion, and others. So we performed similar analysis with 7 different task data from the same subjects, and again performed gradual mode analysis and also ranking. And this ranking is shown here during the task. So here you have different, so this is emotional task, gambling, language, motor, relational, social, and working memory. So the blue circles tells you how cortical functional networks are communicating with the thalamus at resting state, and this color you don't see, so it's a black for you, for me it's red, but so the black is telling you communication during the task. So if the circle is down, that means it has a weaker communication during the task, and if it's up, that means it's a higher communication. And very interestingly, we observed that doesn't matter which task people perform in the HCP sample, still you have exactly representative communication with different cortical functional networks. And this still we have to think more about these results, what does it exactly mean. But this is one of the basically descriptive observation.
So what are the networks that goes down and up? Was the right frontoparietal that goes
So this ordered is basically this order. So frontoparietal, right sensory motor, auditory. So that's the order.
Yeah, I was just not able to remember the exact.
Yeah.
I'm looking at the next slide.
Yeah.
Sorry, could you re-say if it goes down, it means it's higher?
So yeah, if it's up, that means it is higher in the task. If it goes down, that means it has lower communication.
So it's slightly counterintuitive. The frontoparietal, the intraangular frontoparietal, it's disengaged in all the tasks compared to rest, according to these stuff.
Yeah.
Is that correct?
And then the auditory goes up. Why?
Yeah.
So auditory,
Right? Auditory is the one that.
and let me check. I also can't read actually. It's just.
Yeah.
So auditory could actually go, maybe people have a lot of visual auditory instruction maybe during the task probably in the HCP
These
sample.
are all on-off designs.
No, this is the human connectome project design.
The task designs are all on-off.
I have to clearly check, so we just took all the data and just put it just for the tasks only.
'Cause I guess
Just
that's the commonality,
one commonality, yeah. So so if you look, a lot of work people record like electrophysiology with, and then you have that there are specific neurons, for example, in pulvinar for the face, or I think there are some specific areas. So one of the thing could be that fMRI is not specific enough to capture it. Second, maybe that processing to capture the face, which is reported in the like macaque or mouse data, that is just so rapid, like this just really happening on some temporal scale, which is then just doesn't become representative in such a time average correlation analysis maybe. And this basically summarized. So we looked at 10 cortical resting state network, and they showed ordered arrangement in the left and right thalamus. The representation of resting state network varied in size, localization, and laterality differences. This just I skip. And the rest to task transitions seem to involve the thalamus in a unified communication pattern. And before I end, I want to end with one take-home message, and that is that thalamus and cortical connectivity is needed because one cannot live without another. Thank you for your kind attention. Yeah, please.
Can I ask, those results were all done with instantaneous correlations?
Yes.
So
For the parcellation, right?
Right, yeah.
Yeah.
I wonder whether these slightly counterintuitive prima facie, so who knows, maybe there's a much better, maybe they make sense once you look at the exact tasks, which seems like an important thing to do. Like, you're right, all of them are auditory. But could you instead be removing the central, because essentially that's what you're doing, you're removing, right, the central, you're using the big average in order to boost up the little transients. Right, that's the whole point of doing those instantaneous correlations, right? You're really highlighting the transients in order to get clusters that have similar transients.
Yes, yes.
So I wonder whether actually that might, to some extent, explain why some of those counterintuitive findings, like the intraangular, so the frontoparietal being interested in all of these tasks, which feels slightly counter to expectation.
So, extremely interesting. So when we published the other functional parcellation paper, then reviewer asked us to look at these parcels and looked at their coherence. So coherence, like the correlation in the frequency domain. And we were looking, so I just show you one time. I think it's not here, but it's extremely interesting question, extremely interesting observation you paid. So what we were looking is then, sorry, I didn't put in the PowerPoint. So basically here you have right and left parcels, and this is correlation in the frequency domain. So between 0.0 to 0.1 hertz. and you can see like there is higher coherence between different right and left parcels, which makes sense because in resting state operates under 0.1 hertz. But we see that, for example, parcel 9 and 12 have higher coherence, and this higher coherence persists also at coherence in the higher frequency range. So 0.1 to 0.2, 0.1 to 0.3, and so on until here. And this, we were interested, what does this 9 and 12 mean? And this is basically, so that's basically 9 is central lateral and 12 is central median, and that is interlaminar nuclei. And they have wider functional connectivity as well. You don't see it here, but this has all over tracer projections in the mouse brain. And this has been implicated by Brian Edblow from Harvard in subcortical arousal network. So he did DTI in the ALS patients, and he found that there is disrupted connectivity between the brainstem and centrolateral and centromedian in the coma patients. And there has been also some work by SIP showing there is a loss of arousal in lesion in centrolateral. So yes, it could give even more insight if we apply like instantaneous correlation analysis method, even at the cortical network, even by looking at cortical functional network. So it would be even more insightful. So really, very, very nice point.
Yes. A lot of the thalamic nuclei have somatotopic or renotopic or tonotopic organization. And I can imagine when you look at your functional data, two extreme outcomes. One would be that the functional data, because it's not task-driven or event-driven, is just completely blind to that topic, or alternatively that it might, that a nucleus might sort of arbitrarily get split up as you get more and more data. Do you have a sense of which of those might be more correct and what it tells us about the way that we should think about resting state data?
That's extremely interesting point. So so there has been old work by Hustler, and they're showing that there is a nice somatotopy not only in thalamus but also in basal ganglia. And we have looked at this point in more detail. So volunteer perform different motor tasks, and we want to look whether there is some kind of somatotopy in the thalamus. But fMRI resolution is a little bit having issues. Same bold response is more issue because you have a lot of draining vein effect. So we don't see clear somatotopy. But answering the second part, how does functional parcellation could render on to understanding such kind of function? So we are looking, this is why we are looking like this high dimensional functional anatomy. And one of the interesting observation we had is when we looked at in high resolution data, not at 3 Tesla. So when you have better spatial resolution, then you observe some of the interesting patterns. So I can show you one, for example. So like here somewhere. Yeah, maybe this here. Okay, this is just a plot. But this shows like not only thalamus but also basal ganglia. So the kind of reliability score I showed you. So that's basically moreover having very good range actually. So it's not like they are really going up and down. So some are really having peak higher reliability, but others are also not so bad, and that suggests maybe there is some kind of variability. But one of the interesting thing is that it doesn't matter how you chop the fMRI data, you are like having chances that the space is like preferred for the data, and this make a lot of sense looking these kind of parcellation compared to the task fMRI studies. For example, having motor task and then rendering which parcellation makes sense. This I think should be done. It was a really nice question.
James White raises his hand first. Sorry, James.
Just two things I didn't quite get clear. First one was during the visual task.
Yeah.
What were the connections you were looking at in the lateral connected body, in the posterior pulvinar, in the thalamus as a whole? Where were you actually looking? Where were those connections?
So we looked at all over, but so you mean within the thalamus?
Yes.
So within the thalamus, actually there is no, so nuclei doesn't connect with each other. This we know. with the animal work. So they only connect with either subcortical structures or with cortical structures. So what this connectivity picture you saw, that basically tells that thalamus is communicating differently, either with subcortical structures or cortical structures actually. So that's basically the representation there. And saying so, one could exactly hypothesize, let's say you are interested in ventral LGN or lateral LGN or interior LGN, then one could ask different questions and then see where they connect or with which layer in the cortex. So this could be answered with the data.
Just one other thing I'd find. The parcellation within the thalamus related to its connection to the cortex, was that parcellation histologically different?
So histologically, there is a correspondence with the parcellation. I think just a second, I show again. So it's here. So I think you see it here. So there is alignment with the histology, but it's not perfect alignment. So like this is, for example, all parcels in numbers and these are in the letters. So these parcels what you showed, that has a rough correspondence with the histology.
As a
rough correspondence, not like complete match with the histological cluster.
So initially I was going to follow up on Roger's question, and that is you don't have a true ground truth, right? You have the histology here, which is, as you say, an approximation based on a small number of subjects. So at what point, when you go to higher and higher power and get smaller and smaller parcellations, do we say that those parcellations are, I'm really just reiterating Roger's question, right?
Yeah.
At what point do we say this is a nucleus that has this function versus here's how I've subdivided it into many, many things? So where should those of us who are interested in generalities look, at what level?
Yeah, so we looked this, so this was also one of my questions when I looked at float until 50, then I computed until 120 dimensionality. And you end up in moreover linear stability. So I do not know where this is gonna end. I think it's limited with the resolution. Some point it can break down. Secondly, it can be also statistical error, because how you randomly divide, maybe that has some kind of bias because it's like Voronoi diagram. So you could get overlap as you get smaller and smaller. That is another problem. Since we don't have a ground truth, so we do not know exactly, basically.
And the question I kind of wanted to ask was, here you showed a difference in laterality.
Yes.
And you have differences based on the histology here. And then again, you showed differences in laterality later that was functional.
Yes.
How much overlap was there between the differences in laterality? And were they consistent? And if we were to go into our histology atlas and say what areas should we be looking at for the side-to-side differences, what would you recommend?
I would, answering first has to be done. Actually, we didn't compare. Secondly, so I was just now in Gordon conference, and they're not in the talk, but I was personally discussing, someone is labeling the projections of the subcortical structures to the thalamus, and the thalamus basically histology is really, I think it's not reliable. So the projections are still not well understood. So what we are looking at is not really depicting true anatomy. That is the problem. And second is like the atlas we use, for example, Morel. No one knows like how did they draw the boundaries. So it's really, I think there is no understanding about it. And third, inter-individual variability. So if one could make a, let's say, atlas, then how you model that kind of inter-individual differences. So there has been a book by von Beuren in 1973, and he looked at postmortem samples and draw different nuclei boundary. For example, mediodorsal, in I think 14 subjects, 14 samples, and you can see that there is a really lot of variability just in specific nuclei organization. And that is, I think, the major challenge for us, whoever studying subcortical anatomy, not only thalamus but all other areas. So comparing any connectivity or parcellation result with histology doesn't make lot of meaning actually. But but that is this just the way you have to look at it. Otherwise, there is no other way. So, thank you.
So you mentioned that you don't find direct connections between nuclei. So it specifically structural, right? No direct structural connections between thalamic nuclei.
So we, in this paper, in the supplement, we have whole figure showing that there is a connectivity between different thalamic nuclei or parcels. But in the histological literature, there is understanding that they do not, there is a very specific organization of the thalamic projections and they do not communicate. So each nuclei communicates within, but they don't have communication with the next one. So they could communicate actually either via reticular nucleus or via different cortical structures or different subcortical structures like zona incerta or basal ganglia. So there is a lot of organization, but it's just not there, but on the different level, it could come and connect actually.
And then you also, I think you showed a lot of functional connectivity right between these nuclei.
Yeah.
And how much do you think that the connectivity profiles between the thalamic nuclei and the cortex are moderated or mediated by internuclei cortical connectivity?
We didn't look at it, but it's very interesting, I think, analysis to do. Yeah.
So just a couple of things. So is that also true for subfields of the hippocampus? Subfields of the hippocampus, is that also true that these subfields don't communicate, or they do communicate?
The subfields communicate because you have these PT pyramidal neurons and they have nice kind of, so there is a feed forward and feed backward connection in the different layers. So subfield is basically moreover defined layers basically. That's so there is communication, but there is also communication with these layers through thalamus, as well as so indirectly also via basal ganglia and other like cerebellum or brainstem through the thalamus then.
Okay. And just a comment to your question, when you said you showed some functional connectivity between the nuclei, so it's all based on correlations.
Yes.
So it depends on your threshold and your statistical noise. So.
Okay, because one thing I was wondering is maybe to explain that sort of interesting, the paradoxical effect of the association between that thalamic nucleus and the frontal parietal network decreasing with task. Maybe that effect was mediated by another nuclei that took over that function.
Definitely, yeah. That's it's a whole lot of averaging program. So like, so yeah, we missed that kind of then finer observation in that averaging.
Like, could it be? I was just thinking about the frontal parietal being down on the tasks.
Yeah.
Could it be that when you're doing the task, the frontal part is actually being more activated, and so it doesn't require as much input from the thalamus?
Yeah,
I don't know. Like,
it could be also that it's really specific. So PFC or frontoparietal network mainly may be creating some kind of specific representations, and their manifold is just maybe not so much, like the degree of freedom is less, and that could also explain. It doesn't mean that it's not working, it's just having less correlation. It could be that it's just efficiently doing something. But this is
also the direct pathways, frontoparietal are active in the task. Right, but thalamus is more of this modulatory thing, right?
Yes.
So it just could be a totally different state.
Okay, so let's thank Vinod again.
Thank you.